Hello student, myself Dr. M.B. Shende, and this is our second lecture to BSc Part 1, Same First. Student, in first period we have seen about the structure of bacteriophage, T4 bacteriophage, where how the structure of bacteriophage, you know the different types of viruses are there. Here the name bacteriophage itself indicates bacteriophage. Phage means to cause or to eat. So these phage particles or virus particles which eats bacteria, that is called as the bacteriophages. So in last period we have seen all these structures. These T4 bacteriophage have head and tail portions, and head is made up of different proteins, that is called as the capsids. It's contain a DNA, that is called as the viral DNA, and there are base plates and 6 tail fibrils which are helpful. The heads are made up of 50 microns in length. So this is the general structures of bacteriophage, that all the accounts are given in last periods. This is the bacteriophage. Bacteriophage is nothing but the viruses which feeds on bacteria or which eats the bacteria. That is called as bacteriophages. So do not confuse bacteriophages. Bacteriophages means it's a viruses which are eats the bacteria or destroy the bacterial wall or whole bacterial cells. It's called as the bacteriophages. Today's period we see the multiplications of T4 bacteriophages, multiplications or reproductions. How the reproduction is taking place in these viruses, or that is called as the multiplications, or in other words you can say the reproductions or life cycles of T4 bacteriophage. So in T4 bacteriophage, generally the life cycle is completed in two ways. First way is lytic cycles and another way is lysogenic cycles, that we will see in the next period. Lysogenic cycle. In the today's lectures we see only lytic cycles. So lytic cycle in the bacteriophage, it is also called as virulent phase. Why it is called as virulent phase? Because it brings the lysis of bacterial cells. It destroys whole the bacterial cells. That means those virus which cause the lysis of bacterial cell are called as virulent phase, and the lytic cycle is also called as the virulent phase cycles. So you know, in lytic cycle bacterial cell rupture and the end of processes, therefore it is called lytic cycle, and this whole process is completed in the, these are the 5 different steps. 5 different steps are involved in the multiplications of bacteriophage in case of lytic cycles. So what are the different phases? You already studied in 12th level also. What are the different phases involved in the multiplications of viruses? So first phase is adsorption phase, or it is also called as attachment phase. Second one is penetration phase. Third one is eclipse phase. and fourth one is maturation phase, and fifth one is lysis and release of virus particles by breaking the wall of bacteria. So we see thoroughly all these phases. All these phases. First one is adsorption. You know, this is the cell structures of bacteria. Generally, we use here the E. coli structures. E. coli means Escherichia coli is the name of bacteria. Escherichia coli has some rod-shaped structures, short rod-shaped structures. It has, this is the E. coli structures. So in bacterial cells there is some, these green colors, bacterial DNA are present. Bacterial DNA, that is also called as host DNA. This is the host DNA, host cells, and around this bacterial wall I will put it some dots. These dots are nothing but some protein receptors. A specific protein receptors are there present on the wall of bacterium, and the host DNA is important. The cell wall is covered to the all bacterial cells. Some bacterial ribosomes is also there by playing the roles of synthesis of proteins, or it take part in the protein synthesis. So how actual this life cycle is completed? You know, the viral particles having these base plates and sex rags or spirals or a screw-like structure on the base plate, screw-like structure, and the 6 tail fibrils which play a very important role on the bacterial walls. What happens? These virus particles or virons or bacteriophage sat on a receptor sites. There are specific protein receptors where it knows by the tail fibril of the bacteriophage, and it set or fix on wall of bacterium where the protein receptors are there, specific protein receptor which are identified by these viruses, and at that receptor sites the viruses with its rags or a screw-like structure in the base plate and tail fibril fix on bacterial walls. Here fix the tail fibrils on the bacterial walls here and try to break up the walls. So these virus particles try to break up the walls of bacteria, and the tail fibrils here in the second stage secretes some enzymes which breaks the wall of bacteria here and introduce to the viral DNA into the bacterial cells. So here these virus particle introduce its viral DNA, this red color viral DNA. Once the entry of viral DNA is in the bacterial cells, it takes the control of bacterial cell, all the assembly of the bacterial cell. After the entry of this bacterial DNA into the viral DNA into the bacterial cells, it takes whole power or takes control of cells. Once the entry is is in the bacterial cell. The viral DNA takes the control and the bacterial DNA degraded. The degradations of bacterial DNA starts and it has a phase. Messenger RNA is also there. So here the entry is in the bacterial cells, and this head without viral DNA is destroyed, and it is called as, therefore it is called as ghost. It is destroyed and lapse in the process. After that, this virus DNA, you already know the virus cannot be multiplied without any living cells. Outside it remains in inert state. It requires living cell to multiply. So it choose the bacterial cells and introduce its viral DNA into the bacterial cells. So taking the charge of these whole cells along with bacterial DNA, the degradation is take place and viruses starts producing or synthesizing with the help of its messenger RNA, synthesis of viral DNA and some protein coats. It starts its machinery. These virus use the machinery of bacteria, and by using the bacterial machinery it produces its own DNA and protein coats. So here the degradations, head and tail proteins. So heads are prepared here in numerous manner. First head is prepared and viral DNA is prepared and tail portion is prepared also. First head is formed, then tail is formed, and after that DNA is introduced in the head, and after that tail is joining to the head and a huge assembly of viruses is formed. So virus particles, the first head and tails are joined to the heads, and the viral DNA is introduced into the heads. A large number of virus particles are formed. So in this stage here, for the synthesis of viral DNA and protein coat, at least 15 minutes are required. 15 minutes. Within the 15 minutes the process is completed and huge assembly of virus particle is formed. At that time the huge assembly is formed. It creates a pressure against the bacterial wall. At least one cell produces 100 to 200 virus particles, which creates the bacterial wall, and it brings the lysis of bacterial wall. Bacterial wall is burst or is break, and all the virus particles are liberated in the atmosphere. At least 100 to 200 per cell is liberated, and again these liberated particles can infect to new bacterial cells. Hence the life cycle is completed. New virons are formed, and it can be ready to infect a fresh bacterial cells, and the process is repeated. So time required from injection to appearance of these new viruses this phase is called eclipse phase from the infection to the formations of virus particle here, that is called as eclipse phase. And from injections of viral DNA to the bacterial cell to the bursting of or lysis of bacterial cell is called latent period. Do you understand this? Latent periods. So this type the process is completed. You know the structures of E. coli, that is Escherichia coli. It is one of the bacteria which used by the virus particles here. So this is the first adsorptions or attachment stage. Here the virus particles are attached, and here the second stage is penetration stage. Here the virus particles are sat on a wall of the bacterial or bacterium, and it secretes a particular type of enzymes, and it stretch its tail fibril like this, so that the head come near to the wall of bacterium, and then after that it introduces its viral DNA into the bacterial cells, where the synthesis of proteins and DNAs take place. Then after the synthesis of viral DNA and protein, there is a maturation phase where the head and tails are joined and form a new fresh bacteriophages, and after that a huge number of bacteriophages are formed. It exert the pressures on the bacterial walls, and there are some enzymes that is called as lysozymes and hydrolytic enzymes. Lysozymes and hydrolytic enzymes which creates the hole to the bacterial wall. It creates here hole like this, hole to the bacterial walls, and break the bacterial wall so that all the particles are liberated in the atmospheres. So such type these lysis of bacterial wall taking place. So this lysis of bacterial wall is taken by the virulent phase. So it brings the lysis of bacterial cell. Therefore, it is called as a virulent phase, or this stage is also called as virulent cycles or virulent phase cycles. So lytic cycle is very important from examination point of view. It may be asked for 2.5 marks in the examinations. So you go through it. New fresh notes will be you will get soon. You just watch this video, and if you have any difficulty, please ask me. I will give a satisfactory answer to you. So I hope that you must understand it, all these points, and contact me if you have any difficulty. So this is very important topic from examinations point of view, that is multiplications of bacteriophages, lytic cycles. In another period we will take the another method, that is lysogenic cycles, which also taking place in E. coli also. So this is about the lytic cycles. Watch this carefully. Thank you.